Okay, so continuing a little bit more on the previous class where we had this introduction to complex system, there are of course a number of aspects that we uh, kind of skipped over in view of time. So I want to continue a little bit more on that before we continue with another topic. So one aspect we did not really discuss about is diffusion. So, most of this class, of all the previous classes, we've been talking about kinetics, we've been talking about reactions and all the different mechanisms, the orders, etc., etc. But of course, we briefly touched on this when we looked at the Smoluchowski equation, where the diffusion coefficient also played a role in order to predict the rates. But if we look more generally, diffusion is of course a very important phenomenon. Let's say very simplistically speaking, if you have, let's say, a compartment with certain molecules here, and at a certain time, I open that compartment, those molecules will diffuse outwards, right? They will go throughout the box. And after, let's say, a long time, so this would be T0, this would be T, let's say, infinite, you will have an equal distribution of all the molecules in the volume. So that's spreading out, that is diffusion. And as we'll see, that has also an interesting interaction with reactions. So you can have concentration changes at a certain point in space and time, one by diffusion and one by reaction. So let's look at a brief simulation. So I'm going to show you again with Smalldyne how we can set this up. So this is a very simple script using Smalldyne. So we say 3 dimensions, we make a box of size 100. We have 3 different species, red, green, and blue, and they have different diffusion coefficients. So red has a diffusion of 100, green 10, and blue 1. So there's every time an order of magnitude difference between the diffusion rates. This is just to say how large the markers are, the colors, of course, start time, how many molecules you have. So we have thousands of each, and we start, they are all in the center of the box. So at 0, 0, 0, those are the initial coordinates. Okay, so let's see what that looks like. Looks like this. Okay, so I have a rapid spread out of the red molecules. They go very fast because they have the fastest diffusion. Then the green ones are a bit slower and the blue ones are even slower. And you can see that they kind of gradually spread out from their center position from 0, 0, 0, and they go outwards and outwards. And if we would wait a very long time, then this would all equalize over the entire box. Okay. Go back to my screen. Okay, so how can we describe diffusion? The easiest one is called Fick's second law of diffusion, right? Which says that dc dt is the diffusion coefficient dc dx squared. So where t is time, okay, so this is time, and this is length or space. So this is a one-dimensional diffusion equation. So the way to read it is the change in concentration in time. This is the diffusion coefficient. Diffusion coefficient. We saw it before, right? Normally in meters square per second. And then this term is the change in concentration as a function of space. So this is in one dimension. If you want to write it more generally in 3 dimensions or 2 dimensions, people write it in the following way. So they write dc dt equals d, and then we write it like this. So this is if the diffusion constant is really a constant, but sometimes your diffusion constant can be a function of concentration or a spatial dimension. So then you would write it like this. So what is this operator? It basically is the following. Okay. So this would be dc dx plus d2c dy plus d2c dz. Right, so it's just the partial differential equation, right? So that's why we have a delta and we don't have the straight d's, right? So in the normal rate equations, we were writing dc dt with straight d's, which is an ordinary differential equation, right? And now we're writing these deltas, dc dt, because, sorry, again, should be a delta, because time is only one dimension we have to take into account in diffusion, because space is another. So it's a partial differential equation. So those are much harder to solve. So we won't go into that for now, but at least now you know that diffusion is also a term which can change concentration. Okay, so let's look at a very famous example, which is called the Gray-Scott model. Okay, so it has just 2 equations. Okay, so we have species u plus 2v, which gives 3v. So if you look closely at this equation, we've seen this now in a number of examples, you see that the product in this reaction V is also a reactant. So this is what's called an autocatalytic reaction. Right? You can imagine if the reaction starts at a certain total rate, but I increase the amount, the concentration of V, that then the overall reaction rate will go faster, right? Because I have, let's say, a rate constant, K, and I would have K times UV squared. If I increase the amount of V, the concentration, right, then the overall reaction will go faster. So this is a self-catalyzing or autocatalytic reaction. And then we have V that goes to product P. Okay, so if we would do our normal, let's call it zero-dimensional, right? Well, okay, let's not call it zero-dimensional, but let's just call it, if you would have only time as a consideration, then we would say, okay, we have a K1, we have a K2. So we would have du dt, right? Which, let's see where u is. U is consumed in the first reaction, so it would be minus K1 times u times v squared, right? I'm just leaving out these brackets. Okay, but there are these concentration brackets here. Okay. And that's basically it, right? U is only consumed there. And dv dt, it's being consumed but also produced. And so I'm consuming 2 V's in the first reaction and producing 3 V's. So overall, that leads to K1 times U times V squared. And I'm consuming V in the second reaction, so minus K2 times V. So this would be if we have a very well-stirred system. Let's say we would have a beaker or a reactor with very strong stirring. So there's no differences in concentration in space. So we would only consider this time dimension. Okay, so now consider we have not only time, but we have time and, well, space, let's call it. So time and space, and let's keep to a two-dimensional system. So we would have the same chemical rate equations, but we have now the diffusion terms. So we would say du dt would be the diffusion coefficient of u times this, and we would have minus K1 times u times v squared. And the same, so for d, dv dt would be the diffusion coefficient of v delta v plus K1 u v squared minus K2 times v. Okay, so in this case, there's still an element missing. And I'll tell you why. So this would be, let's say, instead of a well-stirred reactor, you would have, for example, a gel reactor. So a very thin layer where all my components are inside and they can diffuse, right? So diffusion terms diffuse and they can react, right? So that's react and that's diffuse. Okay, so the most interesting conditions is if you have reaction, diffusion, but also a feed. So a feed, so let's say time and space, and a feed, which is an influx and outflux in the system. Okay, so now imagine you have a gel reactor. So this is a quite common tool. And we have a compartment on the top, which is stirred. And we have a compartment at the bottom, which is also stirred. Okay, so here's my gel, and I'm flowing in U here and U out. So here I have a constant concentration of U. And at the bottom, I'm just stirring. Okay, so what will happen is here I have an influx of U into my gel, and here I'm removing my waste products. So let's say in this case, we have U being added, and we have V, U, and P that are flowing out of the system. So we need to incorporate those feed terms in our equations. So let's write them again. So we have du dt. So I'm just going to repeat the same part, right? Minus k1 times u times v squared. And now I have this feed term, which I call f times u. So this is just a kind of dimensionless rate constant, which defines the influx of the system. Okay, so this is influx. And I'm also losing u because at the bottom I have the outflux. Okay, so I have minus f. Sorry, this is not times u because this is concentration independent. So this is just a rate of influx. And this is minus f times u. So this would be the outflux. And for v, dv dt is dv, what did I say, plus k1 times u times v squared minus k2 times v. I'm just copying from above. And I just add another term, minus f times v. So again, outflux. Okay, so what people then do, they make it all non-dimensionless, but basically, let's say we set K1 is 1 and K2, we just call it K. Okay, so then the equations you will see in the textbooks would be du dt is du u minus uv squared, right? Plus f1 minus u, and dv dt is dv v plus u times v squared minus f plus k times u. Okay, so this is the, let's say, most famous notation of the Gray-Scott model. So then there are all kinds of ways to implement this. I'm not going to discuss about those. So how to implement numerically, right? So you can do what's called finite difference methods. You can do finite elements, or like we've seen, you could think of Brownian dynamics. Okay, but I won't go into how all of that works right now. I just want to show you the results. So this is a simulator that that actually implements all of these equations. So you have the reaction terms, the diffusion terms, and the feed terms. And depending on the rate constants, you can get different behaviors. And I'll share this website so you can play with it. So in this system, you can have many different pattern formations. So unlike the Belousov-Zhabotinsky reaction I showed you, which shows spiraling and waves in a 2D gel reactor, these systems, so the Gray-Scott system, can actually get steady state patterns. So let's see what that looks like. So this is one mode, which is called solitons. So I'll just click and it will make an initially high concentration. So I click in a couple of spots and you see the appearance of what they're called solitons, so little dots that are propagating. They're kind of dividing, splitting, dividing, and they make the whole system full of these dots. So what's now interesting, so this is a steady state pattern, right? It's not an equilibrium structure because I have a continuous influx of U. I have a continuous outflux of UV and my product. And that gives rise to these patterns. So it's a self-organized pattern and I can try to perturb it, right? So I'm changing in concentration and you see that the system goes back to its most stable steady state, which are these solitons, right? So there are, again, different modes, right? So this one is also very interesting. So let's start with this one. You get, again, a pattern formation. And it takes some time before the system reaches a steady state. And this is now a reaction diffusion pattern, which can be at very large length scales. So keep in mind that U and V would be small molecules that you would be unable to see, but these patterns might be on the micrometer to millimeter scale. So you can actually see them. So many patterns that occur in nature, for example, there are fish that have these kind of patterns on their outsides, which are a result of reaction networks that lead to these kind of patterns. Let's look at another one. Holes. Okay. Okay, so now instead of spots, we have holes and stripes. These are worm-like structures. Okay. So these kind of behaviors can actually be observed in chemical systems. So there have been, for example, the SEMA system has been studied in a gel reactor where we can see these kind of patterns, right? So pulsating solitons, I really like these ones. Okay, so the main point of the story is that reaction and diffusion can have very interesting interplay, especially in systems that have a continuous influx of one of the reactants, which can lead to these kind of non-equilibrium patterns. And they closely resemble some natural systems that are very important. So one system I want to highlight in a bit more detail, I mentioned it in one of the previous classes, is the MIN system. So you're looking at an overview, schematic overview of the processes that are going on. So you see there are different proteins. There's min D and min E. And at the bottom, you see a cell membrane in yellow and green. And you can see that min D can bind to ADP, which is adenosine diphosphate. That's these two little balls, which are the phosphates. And it can be converted to min D bound to ATP, adenosine triphosphate, right? This is a common chemical fuel in your body that drives chemical processes. So min D, once it's bound to ATP, can then bind to the membrane, like shown here, and min E, which is a second protein, can then associate with min D. And then once that is associated, it can split off one phosphate group, right? So you get hydrolysis of ATP to ADP, so it loses a phosphate, and min E unbinds, and min D, still bound to ADP, goes back in the solution, which can then undergo this cycle again. Okay, so let's see what that looks like if we implement it in small dyne. Right, so we'll not go through all of these steps. So basically here, all the diffusion coefficients and rate constants are being defined. Here we define the cell size and a lot of other properties that I'm going to skip. We define the species, so min D bound to ATP, bound to ADP, min E, and the complex of min D and min E. Okay, so this is the initial distribution of the molecules and the colors. Don't worry about all of this. Here, the reactions are defined, and then we run the simulation. So let's just look what that looks like. So I go to the simulation and I run the simulation. So this is an E. coli bacteria, right, which has these elongated cells, and we'll look at what this simulation does. Okay, so you can see on the right side of the cell, you see an accumulation of the min proteins, right? Then this accumulation disappears, and you see an accumulation at the other end of the cell. So what is thought to happen, and I'll show you a real movie of imaging of these E. coli bacteria. Because of these oscillations between the poles, the cell knows exactly where the geometric middle of the cell is, where it forms a ring of min E proteins, and that actually is the trigger for cell division. Okay, so let's look at a real cell movie that shows this process. So this is recorded with a fluorescence microscope. It has 3 overlapping channels. So it has a blue and a yellow channel and an overlapping channel here. So you can see that there's indeed this end-to-end oscillations of the different proteins in the real cell.